Right, the next thing we need to get started on before break is to start looking at processes in the kidneys. And I'm going to give you this link here. Obviously, it's on the PowerPoint that's on Moodle anyway. But it's a really brilliant little summary of the whole process, and it's so simple. And it's on that McGraw-Hill higher education site. They've got loads of animations on there, and it's so simple. It's useful. And we've got these 4 processes. Ultrafiltration we're going to concentrate on first. And that's what's going on in Bowman's capsule and the glomerulus. Selective reabsorption comes further along, because what happens in ultrafiltration is you get so much stuff squeezed out of the blood. It includes a lot of useful things like glucose and water and certain ions. And so they actually have to be taken back up. So selective reabsorption is taking back materials that the body needs. And we'll look at that not in a lot of detail, you know, because it is complex, but we'll look at it in the PCT, the proximal tubule, and we'll then do a bit on secretion, not very much, but the bit we're going to concentrate on is osmoregulation, because it fits in best with this particular, this unit and the way I've written the assessment criteria. But this link here really just puts them all together. So see if that opens up as it should do. It took it a while to load yesterday when we had a look at this. But before we go into the processes, this doesn't include any huge amount of detail. So it's quite a useful starting point. And it simplifies the blood vessel into just a sort of straightforward tube, and the kidney tubule as well. So all right, now if we just click on the begin bit here, we get this. All right, that's a simplified nephron, and I think that's really good. You know, you look at the diagram that you've got with, you know, Bowman's capsule, glomerulus, artery in and out, and all the capillaries and stuff, and then, you know, the wiggly bits and the straight bits, the loopy bits, all that, and you can summarize it to that. And when you look at that, you can see the parts, can't you? However, if you can't see the parts, what you can do is click on these labels over here, and it lights them up for you, look. So the afferent arteriole is going to be the one coming in at the top, isn't it? There. The glomerulus, of course, is there. The efferent arteriole is there. Okay, so now we can come out of the glomerulus, or out of Bowman's capsule. We've then got peritubular capillaries. They're the capillaries that wrap themselves around all the bits and then join up to the renal vein. The only bit that's identified on here specifically now is Bowman's capsule there. The rest is all just lumped together, right? But that's okay. I don't know whether we can, no, it doesn't give us the option of highlighting them separately. And it then gives us the option to animate, I think, what's happening. All right, blood's coming in at the top and going out. Oh, no, we haven't got anything actually. All right, here it comes. All right, the blood comes in from the renal artery. There's filtration into Bowman's capsule. Now, to be absolutely honest, the size of those arrows should probably be the other way around, because there is more material filtered from the blood than remains in it, pretty much. So I would have had a large arrow going into the capsule there, and a smaller one perhaps in the blood vessel, but that's by the by. So that's the first part. That's the first thing we're going to look at in terms of processes. The next stage, if you carry on through, as the filtrate passes through the PCT, the loop of Henle, and the DCT, you get substances reabsorbed from it back into the blood. That's why those capillaries are wrapped around it. So stuff that we need is reabsorbed. We also, however, have secretion of other products. There's things like used up medication, you know, drugs, hormones, getting rid of more urea and that sort of thing. That's actually secretion. and then it finishes off by the blood oh, it didn't finish it off by the blood rejoining the renal vein, but there you go. That's what that little that link is on the PowerPoint there. So nope, that wasn't the version of it that I wanted, was it? But we can do it from there, from the current slide. Right. So these processes, yeah, we're going to look at them in varying degrees of detail, and the first stopping point really is to see what happens in Bowman's capsule. So this is a large version of the diagram. I don't think you've got one that's anything like this. I'm not sure you've got one that mentions these rather weird little cells called podocytes in there. You might have somewhere. I'm not going to ask questions about podocytes. Oh, yes, there are. If you get onto the textbook bits, yeah, there's some podocytes on there. So you see, this is the thing about the materials I've given you. You've got a lot of variety there, and this is why I've included them there. Actually, those two diagrams at the top of that page, it's called page 389 at the bottom right. Those are going to be quite useful as we work through this. So we start off then with our structure of this and the ultrafiltration bit. We'll hopefully only need about 10 minutes for this. This quite nicely shows you how your afferent arteriole is much wider than the efferent. Look, right? So your afferent's the incoming, so that's that wide. And your efferent is more narrow. All right, so the blood in here, in this big, this is your capillary network in here. So I've spoiled it now, look. But the pressure is going to be high as it comes in. So that increase in pressure is going to force liquids out. That's what the blue arrows are on here. The rather peculiar appearance of them, the pink bits in the background here are the blood capillaries that make up the glomerulus. And then Bowman's capsule around the outside. But you can see all this grey stuff on here. These are these odd cells which are labelled down the bottom here. The inner layer of Bowman's capsule, they're described as podocytes. The cytes bit means cells, yeah. Any idea for pod? Well, pod. What? Well, pod actually is it's a bit of a word we find elsewhere. I'm trying to think of a good example, but it means foot. Yeah? So yeah, that's sort of ped and pod are very, very similar. Yeah. So podocytes are cells with little feet. Right? And you can see the little feet, they're these sort of extended bits on them. All right. Now, they don't do anything very special. They're very unusual, but all they really do is support those blood vessels. So they're there for support. And there's also a membrane. It does say over here, basement membrane, see next slide. As it happens, I have got basement membrane on the next slide. But it's just that because you've got huge pressure coming in here, if you only had blood capillaries with just their walls that were a single cell thick, of course, that huge pressure could just burst them. So you have podocytes around the outside to support them. So you've got this high pressure coming in to just, withstand the pressure, really. Sort of, yeah. So it's giving it a sort of extra, an extra layer, isn't it? And then we have this idea of basement membrane actually turns up in a lot of places, but we hardly ever mention it. Basement membrane isn't cellular material. It's like fibrous material like collagen that really supports layers of cells sometimes. You can't just have loose cells and hope they'll stick to each other. Sometimes they need to be supported on a layer of fibrous tissue. So this is the sort of microscopic detail of what happens inside Bowman's capsule. But it's, yeah, I'm not going to ask you to label any diagrams like that. All right. All we now need to do, really, oh, hang on, let's have a look. This one just shows you how they're related to each other, a slightly simpler version of it. That we've got, here's our glomerulus, here are our podocytes, all right, and then we've got, if we take a podocyte and the blood capillary that's closest, you get this sort of effect in the middle. So this podocyte, well, the podocytes themselves are those, all right, the blue things wrapped around here. So what we're looking at in this second diagram is sort of, if we took one of them, all right, that's really what that is. Okay, yeah. So you've got the podocyte cell on the, it's the inside of Bowman's capsule hanging onto the blood capillaries. Does that make sense? Yeah, to support them. If you then take that a bit further, where is the basement membrane in all this? Well, here you'd have this is the capillary wall, right? That's only one cell thick, all right? That's not very substantial at all, is it? Here's the blood cells inside the capillary. Here is our podocyte. They've kept them blue throughout. And then you've got this layer that just provides a bit of another bit of support. All right, so you have this filtration membrane, really, is all we would describe that as. Now, the structure of it, I'm not going to ask you about in the exam anyway, so don't worry about that. But, you know, if this interests you, then, you know, you can obviously read around a bit. You know, if you look at this online, then, or on Moodle, you'll have, you know, you'll know what that's about. Now, what actually goes on, and I have parked that in the wrong place, haven't I? Down at the bottom, it would be back at the top. Just bear with me. So move the recording buttons up to the top again. So what's happening in that setup where you've got high pressure going in? We describe it as hydrostatic pressure. Hydrostatic is to do with the water content. You can't compress water. So the pressure that you get from it is going to provide support in some organisms, but it also provides this pressure that's going to bring about a filtration process that occurs in here. We call it ultrafiltration because it's not just ordinary filtration, it's extreme, if you like, and it's non-selective. The idea of it being non-selective means that it removes water, it removes urea, it removes sodium ions, potassium ions, glucose, amino acids. Now, amongst that lot, some of that's useful, some of it's waste, but it's non-selective, this process. The kidneys do not just filter out the waste to start with. They filter out everything that's small enough. That's the determining factor. So, I'm just going to check here. Do I ask you the question, what's not forced out? And it's down there at the bottom. You can only force out particles that are small enough, so you don't get cells or protein forced out in the kidney. Hence, if you get protein or blood in the urine, it's a bad sign, isn't it? Yeah? So we know that's an indication that this might be damaged. So the capillary, we know that capillary walls are only one cell thick, and there's going to be little gaps between those cells. So you can describe those as pores. So it's possible for materials to pass through the walls of capillaries because they are just these single cells fitting together, and, you know, to actually get molecules moving between them is not difficult. The basement membrane provides a sort of filtration membrane, and these podocytes, they're not doing anything particularly. They're just holding everything together. So this huge pressure situation is not going to damage the blood vessels. This idea of it being similar to tissue fluid is a bit more extreme. Because, you know, with tissue fluid, you've not got the massive difference in pressure, but the way that tissue fluid is released in the capillary networks, we did that last week, didn't we? We were talking about the incoming pressure being high enough to force tissue fluid out. Okay, now you can see people scribbling this down. You don't need to. I'm going to just move on. I'll come back to this if you need me to. All right, but I'm going to move on to the next bit. The force that actually produces the the filtrate here, similar to tissue fluid, obviously, you can actually calculate it, and it's a result of adding up all the pressure that's forcing fluid out and taking away from that any pressure that might be trying to force fluid back in. And there are three component parts to this, you can see. You've got glomerular blood pressure, the hydrostatic pressure, the purpley coloured arrows on here, which are trying to force fluid out of those capillaries. So that's not all we've got, however. We also have, and this partly results from forcing stuff out of the blood, we've got what's described here as blood colloid osmotic pressure. And what that is the water potential, if you like. It's the water potential created by leaving the proteins behind in the blood. So the blood has a lowish water potential that will actually, if you look at the yellow arrows, try and draw fluid back in again. All right, so we've already got two opposing pressures. You've got the actual blood pressure trying to push fluid out, but you've got the osmotic pressure trying to draw fluid back in. You also, of course, have fluid that's being squeezed out in here will exert a pressure in its own right as well. So you have capsular hydrostatic pressure. Now these, under normal circumstances, if your kidney's functioning properly, have these values, that the glomerular blood pressure, the hydrostatic pressure there, you've got 55 millimeters of mercury. The osmotic pressure, the the water potential, if you like, in the blood is about 30, and then the pressure in the capsule is about 15. So overall, your net outward pressure here is 55 going out, but minus the 30 and minus the 15 that's trying to go back in. Does that make sense? So you actually end up with a filtration pressure of 10 millimeters of mercury. It's not huge, but it's enough to actually force a lot of stuff out of the blood. All right, that's just a standard calculation for that. So your overall, your net filtration pressure is the outward pressure of the blood minus the inward pressures. Now, obviously, if you've got low blood pressure, really low blood pressure, let's say, not healthy low blood pressure. Let's imagine you'd lost blood in an accident or something like that, and your blood pressure dropped. Which one of those is it going to affect? Yeah, well, no, it's going to affect the first one, isn't it? Yeah. So if you were to lose blood, this one's going to drop because you've got less blood. You'd still have the same substances in the blood, so the other, the water potential one, the yellow arrow, would be pretty much the same, and you would probably still have some fluid in the capsule, so that would be similar. But the one that would have the biggest, that blood loss would have the biggest impact on would be your blood pressure. So if that dropped, let's say to, 40, we've got a problem, haven't we? If the others stayed the same, we wouldn't have enough pressure for the kidneys to work. So you need a significant pressure in your blood to make your kidneys work. All right, is what I'm trying to say there. You need a healthy blood pressure for your kidneys to work. Sometimes if somebody's lost a lot of blood, their kidneys will start to fail and they will need dialysis. All right, usually you can get the blood pressure back up again reasonably quickly, but that can be an issue. And the opposite problem, if you've got really high blood pressure, it can actually force more out here than would be healthy, and it can damage these filtration membranes in here. So high blood pressure can often be linked with things like protein in the urine, and there's a particular situation where that is an obvious link, and you might know eclampsia, preeclampsia. Yeah, eclampsia in pregnancy you have high blood pressure and protein in the urine, and that's part of that. If your blood pressure rises, and that it can damage your kidneys, it can be very damaging. So the one they can detect it obviously from your blood pressure, but once it starts putting protein into your urine, then the risk is kidney damage, and then the impact of that obviously is greater, isn't it? So we'll have I think we'll have a break at this point and then come back and look at glomerular function, glomerular filtration rates you can measure and stuff like that, and we'll take it on from there. You'll know